All right, can you see that
We can, and we can
opening slide?
we can see that, and we can hear you. It's
Okay,
the
perfect.
wonder of modern technology. So I'll just give you a quick intro, Ryan.
Sure.
I'm sure I don't actually need to introduce Dr. Carnegie to anyone, but uh, as I mentioned about our colleagues in the US who have been so helpful and open and informative, Ryan has been there from the get-go. In fact, Ryan spoke at a workshop that we had back in 2013 around MSX. So we've been bothering him for over a decade now with our questions about this stuff. And Vims down in Virginia also hosted a mission from PEI of growers and fishers and processors and government folk that went down there in January. So, again, they have been so helpful. So anyway, Dr. Ryan Carnegie here to open our talks. Thank you, Ryan.
Hey, thank you, Peter, and good morning, everyone. I'm sorry I'm not able to be there in person, but I'm teaching this semester, including a little bit later this morning. So I'll be around most of the day, except from when I have to drop out for class. What I thought I would do with the time that I have this morning is to present something of a primer on MSX and dermo diseases. I had asked my Canadian colleagues with the PEI government and Peter what might be helpful, and they thought sort of providing some general background might be of use, as well as addressing some of the important questions that are being posed to to the scientific community, the regulators, to Peter, and so on about what might be going on there and what we might infer based on the U.S. experience. So I will dive right in with that. First, I thought I would just provide a short introduction to sort of the methods we use, because I'm showing some images throughout this presentation, and it occurred to me that maybe not everybody is familiar with sort of how we do what we do and what kind of processes we're using to sort of do these diagnostics, produce these images. So just to orient you, I'll provide a little bit of perspective on that. First, histopathology is a very old method that is still highly relevant to working in mollusk diseases today. And this is basically the production of stained glass microscope slides from basically cross sections, and maybe I hope you can see my cursor here, cross section sort of in the upper middle of the image, a stake that we would take through the middle part of the body of an oyster. We would fix it, ultimately process it for embedding in paraffin wax, cut thin sections, place them on glass microscope slides, stain them, and then we could evaluate that microscopically using these sort of general biological stains, hematoxylin, niacin, to look for any pathogens that might be present, as well as tissue abnormalities and so on. And this is a really powerful method, not only for diagnostics. It's not the most sensitive method for detecting very small pathogens like dermo, but it's a good way of getting insight into the nature of interactions between the disease agents and the host as they may be changing over time and space. We do a lot of work in molecular diagnostics. Of course, we do PCR and quantitative PCR. We also love to do in situ hybridization, which is what you're looking at here. This is basically histopathology, those glass microscope slides like I showed you just a minute ago. But in this case, we're staining not with these sort of general biological stains, which stain everything pink and purple in our sort of routine application. Here we're staining with DNA probes that are specific for the pathogens of concern. And what you're seeing here is all of those green Cheerios are Purkinje's or dermo cells that are primarily infecting the digestive epithelium of an oyster host in this case. So this is a powerful method for gaining insight, in some cases, better insight into the nature of host pathogen interactions and determining in more definitive terms that what we're looking at in a tissue section is, in fact, a specific pathogen. And you'll see a couple images of dermo later. This raised fluid thioglycolate method is a very old method developed in the early 1950s by a guy named Sammy Ray, who was working in Texas. This was an attempt at in vitro culture of dermo that failed, But what it did was basically allow for, the short-term culture, sort of terminal culture of Purkinje cells in this culture medium, these culture tubes. We would put a little bit of gill or mantle and some rectum, which you see highlighted at the left of the image, in this fluid thioglycolate culture. The parasite will enlarge in the culture. After 5 to 7 days, we could take that mass of decomposing oyster tissue out of the tube, chop it up with some Lugol's iodine on a glass slide. And the Purkinje cells specifically have enlarged and they stain black with the Lugol's iodine. So we could see them and quantify them very easily using microscopy. This is, it's an old method. It's actually, though, approachable to a lot of laboratories. It's probably equivalent in sensitivity to the PCR assays that are typically favored, and it's still a workhorse diagnostic assay for a lot of applications. So with that, I'm going to move on to some of the specifics on the parasites, and we'll talk about Purkinje first. So dermo disease is caused by Purkinje marinus, and I have a Purkinje cell highlighted over on the left here, and then this, classic sort of emaciated presentation that we see in the right image with the very thin, watery oyster over on the right, this shrunken body, characteristic of this wasting disease caused by dermo. This is a native parasite to the eastern and southern U.S., at least, in historical terms, as far north as the Chesapeake Bay region, though it occurs much more widely north into Maine in the U.S. these days. And of course, it's been found recently in the Atlantic provinces. This has been the most destructive disease of the eastern U.S. oyster population since the mid-1980s. And what we're looking at here, so this is some of this histology, is the disease caused by this parasite, where you can see what normal digestive epithelium looks like to the upper right of the image, this nice tall columnar epithelial tissue. And then moving around counterclockwise to the left, you can see this sort of more lacy, ravaged epithelium. So this is the stomach lining that's being destroyed by the presence and the proliferation of Purkinje marinus cells, which is sort of a typical presentation of what we see in U.S. oyster populations these days. Purkinje is a very prevalent pathogen in our populations. This is just a quick grab of one graph of dermo disease in the Piankatank River. So this is one of our monitoring sites that we study every year. We look at 3 oyster reefs in the fall, which is peak dermo season in this particular river. And you're looking at prevalence on the y-axis. And, this is the average of those 3 sites with standard errors. The average will jump around, but it's always quite high. Sometimes as high as 100% at all 3 sites. Sometimes it's a bit lower, but it is always very high. Dermo Purkinje is a very, very prevalent pathogen and disease in our populations. It probably causes only 20 to 30% annual mortality these days, which is quite a bit lower than it was causing during its peak in the 1980s. What you're looking at here is box counts that were conducted. This is some 2017 data at a reef in the Rappahannock River, which is one of our hotspots for dermo in the Chesapeake. And you can see the box counts. So boxes, these are the cluckers or the clappers, the empty shells that are still articulated at the hinge. So basically, if an oyster has died and it's still articulated at the hinge, you could basically assume that oyster has died sometime within the last year, not probably much before that. So it basically integrates mortality over the last season. And we could see that mortality peaking in the intermediate size categories of the oysters. So this is box counts relative to live counts. We can get our mortality percentage. It's lower in the lowest, the smallest size categories, which are youngest and haven't really been exposed much to dermo. It's largest, or it's also low in the very largest oysters, a size category and age category that probably is enriched for resistant animals. But overall, at peak, probably something like 30-35% mortality. And 2017 was a pretty strong year for dermo disease. So this is probably a maximum level of mortality in that reef. Some fundamentals on Perkinsus, on dermo. This is a directly transmissible pathogen spreading directly from oyster to oyster in a population. It can spread via the deaths of infected hosts, releasing the parasite to the environment. Also, to some extent, through feces. You saw in an earlier slide all of that Perkinsus that was infecting the stomach wall of the animal. So, in life, before these animals have died, and they may be infected for a number of years before they do die, they could be sloughing off parasite cells into the environment with their sort of defecation, on reefs and in farms. Infection impacts by dermo in our waters tend to peak in the second or third year of exposure. So we see sort of minimal impacts in the first summer of exposure, but they sort of gradually increase year by year after that. The seasonal peak in infection is generally August to October, which is also when mortality tends to peak. And infections tend to reach a minimum in the late winter to early spring, when in many locations the parasite can't even be detected at all. With warming temperatures in the spring, the parasite will start proliferating within those infected hosts, in which it's overwintering at very low prevalence, or intensities rather, and it will become more easily detectable and start to cause disease in oyster populations. Temperatures above 20 degrees Celsius tend to favor the parasite, especially above 25 degrees. That's when it can proliferate at rates that the host might have trouble controlling. In terms of salinity, full activity of the parasite, we can see at salinities generally above 10 to 12, though Perkinsus is tolerant of lower salinities. And unlike MSX, which I'll talk about in a minute, Perkinsus or dermo is not sort of knocked out of oyster populations by freshets or low salinity events. It will persist at lower salinities until the salinities become more favorable. The secret of the oyster success with dermo in our system is really disease resistance and tolerance, which we see abundant evidence for both of these. Resistance, basically, the parasite is very prevalent in oyster populations, whether they've been exposed for long or not, whether they're always in higher salinities or in reasonably lower salinities. But what resistance does is tend to sort of moderate the maximum infection intensity levels that the oysters experience. Resistance tends to sort of take the top of the peaks off and prevent the infections from becoming very, very intense and lethal, in very large numbers anyway. I mean, again, mortality is still about 30% annually, which is not insignificant, but resistance has sort of prevented that from being 70 or 80% mortality by not letting as many infections get to those very, very high body burdens. Tolerance is the other part of the story with dermo, and what you're looking at here, this is two images of oyster reproduction. On the right, you can see this is basically a contemporary sample. This is what oysters look like in our system today. Lots of egg production. These oysters are all infected with Perkinsus, but most of them are not so heavily infected that they just can't normally and naturally go through their normal reproductive processes, in the wild, or if they're diploids in aquaculture, producing a lot of gametes and just happily reproducing. Over on the left is what they looked like early on in the period when Perkinsus was at its most intense in our region, where these oysters were really struggling to be able to put the energy into reproduction. You see very, very few gametes, female eggs in this case, in this image, because the oysters just were intolerant of the presence of any dermo at that point, and they were incapable of really doing much of anything reproductively. Today, because of tolerance evolution, they're doing much better at reproducing despite the presence of the parasite. The key question with regard to Perkinsus is what's going on that's important ecologically, evolutionarily, that we can't quite see. We know that there's a lot of questions about the ecology of these diseases that we don't understand, even after many decades of study. With Perkinsus, especially what's going on outside of the host. We know that they produce these resistant hypnospore forms. This is the stage that's produced and stains black in these RFTM, raised fluid thioglycolate cultures. On the right, we're looking at a hypnospore stained from an environmental, basically a mud benthic sample from the environment, using in situ hybridization with specific DNA probes for the parasite. We know we could find these forms in the environment. There's a zoospore of Perkinsus. This is the form that's produced from the hypnospores. It's on the left of the slide here. These are environmental forms that we can detect. We know they're probably important in terms of the epidemiology of the disease and infection acquisition in these systems, but we don't really understand how or to what extent, to what magnitude, as we tend to sort of focus our thinking only on the direct oyster-to-oyster transmission dynamics. There's probably infection acquisition that's happening from benthic stores, like imagine like seed beds or whatever of the parasite, that is probably consequential too. We just don't have a good handle on to what extent that's the case. So let me move on to H. nelsoni. This is the other major parasite, and of course that's the primary focus of concerns in PEI these days. It's another protozoan parasite. Unlike dermo, this one's exotic, was introduced to Asia or from Asia sometime prior to the 1950s, emerging in Delaware Bay in 1957, Chesapeake Bay in 1959. And here I have in some histology, this is sort of, kind of what we routinely see for MSX disease is this initial infection often in the gills, and then this massive proliferation of the parasite. You can see I've got labeling here, highlighted kind of where the parasite is. Massive proliferation of the parasite in the gills, which eventually becomes systemic through the animal. Massive infiltration of host cells into that organ, and this sort of battle on that kind of gill battlefront between the host and the pathogen that can produce a lot of sort of stress and host damage, and so on. This is sort of part of the early stages of dermo disease or MSX disease, and you can sort of even just appreciate looking at the image, comparing it to normal gill on the left, how stressful and potentially lethal ultimately this could be for oyster hosts. Unlike dermo rather, MSX tends to not be very highly prevalent in our systems where there's significant resistance to MSX that's evolved. I've got a couple of snapshots from a couple reefs here from our fall survey. MSX can be undetectable in many years. When it is detectable, the prevalences are generally rather low. You see in the Haney bar graph, this is sort of a couple of, three actually minor outbreaks that you could see pass that probably produced a little bit of mortality, but generally not dramatic effects in our system. So some fundamentals: MSX is not directly transmissible among oyster hosts. Infection and acute impacts are possible, not just on the largest, oldest oysters, but on all ages and sizes, which is really an important distinction versus Perkinsus or dermo. We can see MSX impacts on even the youngest and smallest seed. MSX will develop much more rapidly in oyster populations than dermo will, potentially killing within 2 or 3 months if oyster populations are susceptible. We see seasonal peaks in the spring and fall, generally not in peak summer, not in the depths of winter. But these sort of the timing of these seasonal outbreaks certainly can vary with geography, in the colder northern waters versus the southeastern subtropical U.S. And it also varies in part depending on when a certain region might get most of its freshwater input. If there's a lot of freshwater input in the spring, there may be a minimal spring peak and maybe a more serious fall peak in MSX disease. MSX, at least in the U.S., we tend to think it's limited only by very low temperatures, maybe below 5 degrees Celsius. Though I know the experience of the Maritimes is sort of having us rethink whether MSX is limited by low winter temperatures at all. In terms of salinity, above say 12 to 15 parts per thousand is where the activity of MSX is greatest. It's intolerant of salinities below 10. So unlike dermo, MSX can be completely pushed out of oyster populations by these low salinity events. The secret of the oyster's success with MSX in our area is disease resistance. And this is a graph that, I don't want to linger on here too long because it's a little hard to explain. And I probably should have thought better of putting this one in the presentation. But if you just look at the right side of the image and the divergence between the green and the blue and white trends there, the green is naive oysters that we look at every year in our York River. And the white and blue that's wild oysters on a natural reef nearby. This is maximum MSX prevalence, as determined annually across these sort of very well-studied sort of oyster, deployments or reefs or whatever. A lot more disease in the naive sentinels than in the wild oysters, and the disease and the MSX infection prevalence in the wild oysters getting lower and lower over time. And that's really the divergence between those two trends is indicative of disease resistance. We have other evidence for that as well. The key question with regard to H. nelsoni, of course, is what is the intermediate host? And we, like others, are trying to address that question every day. So some questions that were passed along to me that Peter's been hearing, which I thought I would sort of finish the presentation sort of addressing, since these are on your mind. First, about disease progression. One question that Peter forwarded to me related to whether pre-existing infections were really what's important in driving sort of infection growth in an oyster population, or are the sort of new infections acquired from the environment or from other infected oyster hosts more important? So is it development of the parasite of MSX in this case, or dermo within the oyster, or an infection acquisition from the environment that's more important? This is a good question that we don't have certainty as to the answer of. With dermo, I'm tempted to say that pre-existing infections are maybe more important. The oysters that are dying of dermo disease, have been exposed for a few years. They've carried overwintering infections from at least 1 year or maybe 2 before these infections are proliferating to lethal intensities. It's what probably is important is whether or not an infection that's already acquired is going to develop and proliferate within the oyster tissues fast enough and early enough that it can reach a lethal intensity before cold winter temperatures swing the balance and the dynamic back in the oyster's favor and allow it to overcome the infections. Infections that really don't develop because of maybe lower salinities or cooler water conditions until August are never going to become intense enough to kill those oysters. Those oysters are going to be saved by falling water temperatures in the fall. But if we have a very, very warm spring with high salinities, where the parasite in these overwintering infections are able to kind of roar out of the winter, very quickly, that leaves a long period of warmer summer temperatures for infections to proliferate and reach high or lethal levels. So we might expect to see more mortality in those cases. So that's my guess with regard to dermo, is that the pre-existing conditions are more important maybe than what's acquired from the environment. I mean, that's a hypothesis that we should try to figure out how to test, really. But that's kind of what I think is going on. With MSX, it's probably new infections that are acquired from environmental sources that are more important. Because a lot of oysters are not infected with MSX at all, and then all of a sudden they are, and they can develop very quickly. Another question, is dermo a significant threat to PEI, or really is MSX the story there? And this is a tough one. We look at Maine, and I know that you're looking at Maine and trying to think about what all that means with regard to what the maritime provinces might be, expected to experience. We know that Perkinsus is present in Maine. It's widespread in Maine oyster populations, and we have very little, we've seen it very seldom in the microscopy. So we have a very hard time detecting, what might be considered clinical infections that we could sort of view, visually and appreciate the disease. It's really only just this, PCR detection of low intensity infections that may not be doing much of anything with regard to the hosts. So that's kind of what we see in Maine. If dermo is new to PEI or Nova Scotia, it may be countering naive oyster populations, which you conceivably would have no inherent resistance and could be devastated by the parasite. But we don't know that that's really the case, and that the parasite hasn't been bumping around at a very low level in cooler Canadian waters in the same way that it may have been bumping around at a low level in Maine, causing just no pathology at all, functioning very differently than it does in the South, kind of transmitting itself through low intensity infections without requiring high levels of oyster mortality to kind of distribute the parasite to new hosts. I present this image here. I should have asked Dave Groman if he minded if I presented this. But this is some of the slide in the upper part of the image. This was some confirmatory analysis that we did using in situ hybridization to look at MSX in a Canadian oyster rather, in which David observed it. At the bottom image, this is a control oyster from Virginia. And it doesn't take a whole lot of expertise in histopathology to see that the dermo cells look a bit different as they're stained green in the upper image versus the lower image. This is just, this is one oyster, and we need to do a lot more work to understand what dermo is doing up in Canada. But I'm not looking at this and seeing evidence that, well, the Canadian dermo is just the US dermo from the mid-Atlantic that has somehow found its way up to the Maritimes and is just, hitting oysters hard up there. It may be that this is sort of, a different Perkinsus, maybe genetically, maybe ecologically, that's just doing different things. It's Perkinsus marinus, but that's not to say that it's sort of the same Perkinsus marinus doing exactly what it's doing in the mid-Atlantic. And, if it's been around for a while, maybe it is the case that Canadian oysters have developed some level of adaptation to it. That's, sort of optimistic and hopeful, but I wouldn't necessarily assume that dermo is going to be devastating in the near term to Canadian interests. I could be proven wrong, but we have no concern about what's going on with dermo in Maine because we're just seeing nothing in the way of disease and mortality caused by this parasite there. And finally, parasite burdens in resistant oysters. So what do resistant oysters look like? And, we've been challenged on this forever in the US. I would have growers that would be like, look, I don't want to hear you talk about resistance and resistance evolution until you show me a dermo-proof oyster or an MSX-proof oyster. And that just sort of misses the point on how the evolution of these interactions is working, and certainly with regard to dermo, where we might expect these relationships to land. For MSX, we may get closer to an MSX proof oyster because the resistance, MSX resistance tends to manifest itself in the prevention of the establishment of any infections at all. Resistant populations resistant to MSX tend to have very low or undetectable prevalences of MSX. So MSX resistance is sort of expressed at not letting the parasite gain access to the soft tissues of the oysters at all. That's not absolute, but prevalences tend to be very minimal in resistant populations when they've sort of had enough time to evolve to that point. With dermo, it's a different story. Even resistant oysters can be infected with dermo at a very, very high prevalence. And what I've got here, this is some recent data where we've been working with the aquaculture industry to look at dermo infection levels, Perkinsus infection levels in tetraploid oysters, triploid oysters. Diploid oysters would be no different, but this is just what they're growing. Basically year one, so these are oysters that are only like 6 months old, versus year two, which are oysters that are something like 18 months old. I'm looking at the left panel here, and you can see the prevalence across all the farms that we were studying um, of the parasite in its sort of first year of exposure versus second year of exposure. Over moving to the right of the same image, the same panel, wild adults from nearby wild oyster reefs had generally higher levels of dermo infection. And then the naive sentinels, the truly, unadapted, you know, susceptible oysters have significantly higher levels of prevalence still. So we see that there's a lot of prevalence in wild oysters that have even been adapted, but it's a bit less than naive transplants, but far lower prevalence in aquaculture farms. The real story, and it looks like I had some of the graph cut off here, but it's the same sort of organization. The real story is that resistant populations in the wild and in aquaculture, even if they're infected, they have very few of the advanced infections. So this is what I said earlier in the presentation. The resistant oysters are doing a better job of keeping infection levels to tolerable levels, not letting as many infections reach very high and lethal intensities. And then, of course, I mentioned earlier that tolerance is the really important part of the story, and another important part of the story, where even with these infections, um, adapted oysters, quote, resistant oysters, are doing a better job of being able to manage all of their normal physiological processes despite dermo infection. So I'm going to leave it at that. I know I'm about at a half an hour here, and I'll be able to take questions later on. I'll thank you for your attention. Thank you for having me once again to your group, and I'll look forward to being here for the rest of the day to interact with you. Thank you.
Ryan, we are actually running a few minutes ahead, so we can just take a couple of questions just for you, if there are any. They obviously haven't woken up yet. Jimmy, you'll need to use the mic. Don't ask me, I can barely see you.
Need new glasses. Hi, Ryan. Jimmy.
Hi there.
I have a question. If you were trying to get some resistance in your oyster, if you're starting off, instead of breeding like one male or 10 males with 100 females, wouldn't it be better off going one-on-one? Because we don't know which one for F1 for the first family.
Mm-hmm.
Because you don't know what's at the best you're going to get 25% that will have any resistance. Is that right?
Yeah. I mean, I think I would maybe tend to agree with you. And I mean, this is not my expertise, right?
No.
But I know where the professional breeding programs are often tending to go, and it's sort of this family-based breeding with, I mean, maybe it's one female and one male to produce a family, and then they can produce a lot of families and look at who's performing well versus who's not performing well, and then constitute the lines based on those families. And I think the modeling may suggest that is a good way to get moving along the pathway toward resistance, fairly rapidly. Because as you say, you could sort of identify the individuals that have the right set of genes, behind the high performance phenotype, rather than having sort of the noise that's inherent in, kind of a gemisch of, some number of parents, where you can't resolve it as clearly. But, that being said, even if, and I may not be fully accurate on that, even if, this family-based breeding, is the ideal, there is, I think we do need to, we're limited sometimes by, by resources, by space in hatcheries, by, the funding that we can use to run breeding programs and so on. So, it is a matter of doing the absolute best we can with the resources that we have at a given point in time. And sometimes, we're limited in the rate at which we can move forward because of resourcing.
Wouldn't it pay off also if you raise them to 3 inches? If your market is at 3 inches, before you bred them again, say you have the first family, breed them you wouldn't breed them until they got to 3 inches. That way you would be able to know who has some resistance if they're exposed to MSX and dermo.
Yeah, yeah. Well, I agree with you on that, and that's been, I think, one of the things that we've been thinking of, even in our breeding program here, that, if we're making calls, too early on, who the high performers are, they might not have received the
Resistance.
challenge that would allow us to really identify who is a high performer. We may think we're making good calls, but we may not be. And so that, I think, would be a consideration, is however breeding is being done, that if we're identifying high performers, we need to have assurances that they are. And I think our MSX problems in recent years here have been because we've been making assumptions about high performers with regard to MSX disease that have not been valid. And we've seen an erosion of MSX resistant breeding lines because of that.
Okay, thank you.
Yep.
Jason.
Hi, Ryan. Thanks ever so much for your presentation. Really enjoyed it. The slide you showed that was quite noisy, I think you were showing the naive versus the resistant strains there. There seemed to be a period between 1986 and 1996 where the resistance, or there's higher mortalities in the more resistant oysters. What was the cause for that? Do you have any?
Well, so what you looked at in that complex trend was there was a long. If you think about what was happening, and I probably don't have time to pull this up again, but the left side of the graph, there was an initial colonization of that wreck shoal oyster population, that wild reef, by the parasite during the early MSX outbreaks of the 50s and 60s, and then MSX was gone from that population mostly, and that white and blue trend was bumping along at basically zero. And then there was this period from the late 80s into the early 90s where the two trends were exactly sort of parallel and overlapping before that period of divergence. And our interpretation of that is that period in the 80s and 90s, the wild oysters at wreck shoal were basically, they were unselected, non-adapted, and equivalent in susceptibility to the naive sentinels in our green spring imports deployment. And it was only after maybe 2 or 3 oyster generations by the mid 90s that those trends started to diverge, and the resistant oysters were displaying improved performance in the wild as manifested in reduced maximum prevalences of MSX infection. So that's what was going on there.
Thanks, Ryan.
Yep.
A couple of questions in the chat, but I think we'll save those for the Q&A panel at the end, or if you're on there, if you're able to join us again for the Q&A panel, that would be wonderful. Okay, thank you so much, Dr. Carnegie.
My pleasure. Thank you.